That's a park that's close to my heart and to where I grew up. When I sift the leaf litter on the eastern end of the park, it smells like home. Because that's, of course, what you do when you go to a provincial park. You sift the leaf litter. Don't try and tell me differently. Okay, well done. First thing I want to do is say, well done you. I did not think, I was not sure how well this letter search in a complicated image would go, but well done you. So, I think you found all 13. Did anyone unscramble the word? Anyone? Bueller? Put it in the chat if you think so. Anyone? Well, first of all, so congratulate yourself, pat yourself on the head, and say well done, because those 13 letters were very hard to find. Let me save that, clear all, and there they are. So I think you got all of them. Maybe not that I in the bottom. I don't remember that. I'll go back and look at it on the video. But it's mitochondrial. That is the word of the day, and it's not specifically a taxon word. It's not a word about a particular species, but it's a word about an organelle that we talked about way back in January when we were talking about the serial endosymbiotic theory in terms of the evolution of eukaryotes, multicellular life. And something that we've progressed throughout the term to actually sequencing DNA of the mitochondria of things that live on your face. And I'm excited because that's what we're going to talk about today, is the results of your facial mite survey. And we're going to do more than that. More. We're going to talk about your results in particular, but we're going to combine them with the results from those people, those lesser types from the years 2019 and 2020, who we also sampled in this way, or in a similar way, let's say. You had some additional hurdles in your mite sampling this year that you all navigated very, very well. So, first off, keep letting people in. What we're going to do is talk about this project. Who lives on your face? So you remember these. These are your facial mites. And way back on the 7th lecture, if you can remember, I think that was like the 20-something of January. It was cold. It was more cold than today. How much more cold? Today I'm going to open the window. So the window's open, and we're going to talk about your mite data. So these are Demodex Follicularum. You can see this one pedaling away, and if you count the number of appendages there, paired on each side, you know that this is an arachnid, right? This is an arthropod. This is a molting thing that has 8 legs, and so this is one of those groups of arachnids that we talked about when we talked about the chelicerates and the euchelicerates. And these are ones that live on you, on all of you. So when we start talking about the results, just in case you don't remember the 25th of January, because I don't, we're going to go through and we're going to kind of remind you about the beautiful things that you share space with. This particular bicycling one I like on the right hand side, because that's one that lived on me and made me feel positive about myself, because it meant that I wasn't a barren wasteland of biodiversity, that I too created habitat for something. Well done me and my follicle-free head. So, Demodex in review. This is a reminder, we have basically 2 species of Demodex facial mite on us. We have Demodex Follicularum and Demodex Brevis, and they live in hair follicles, associated with hair follicles. That's the longer Demodex Follicularum, and slightly deeper tucked away into sebaceous glands, Demodex Brevis. And they look, Brevis is called Brevis because it's kind of shorter, and it lives in the sebaceous glands, and they're a little bit more solitary. We find more examples of Follicularum living in groups, and we know also that they tend to migrate. What does that mean, they migrate? Well, it means when you're sleeping, they come out at night and they kind of move across your face. And why would they be doing that? Why, for sex, of course. So, These are the things that you create space and you create food for, and you are even the bar scene where from one follicle to another they're going to go around and look at who they're going to procreate with. I remember bar scenes. Like pre-COVID? Sure. And we've known about these things for a long time, which is kind of amazing, but we're still discovering things about them, which is what you are literally contributing to in this project. So, the first writing actually was probably about these mites was probably done by an Aztec in 1552. And then Europeans caught up with Aztecs later on in the 1840s, several hundred years later. Demodex literally means fat worm in Greek, and follicularum just means it helps you be reminded that they're associated with hair follicles. And we found them again and again, different kind of branches of European science, and medicine in particular found them. And they rediscovered them particularly on corpses, and they particularly noted, or they were surprised to note, that they kept living once the corpses were dead. And weirdly, these species, these two of them, were only split in 1972, which I will insist is not that long ago because it's the year that I split into this world. So, but they're genetically super divergent, and we know ecologically they live in very different niches on your skin. So we know that they tend to, you don't kind of come out and arrive into the world with them, that they're acquired shortly after birth, and so there's a very intimate association that likely the mites that colonized you received from your parents. So mother and infant transmission during nursing is really important. And we also know that as your skin, as the kind of the habitat that you offer in a mite changes, you become a better mite habitat as you get older and as you get greasier. So there was one estimate in one particular study that 95% of people, let's say 95% of people who are eligible for a COVID vaccine right now in this silly province of Ontario, probably have Demodex. Now that number is variable, and likely we all have this. So this is one of the very first things to remember, is that this Demodex Follicularum and Demodex Brevis, these are a normal part of a healthy skin ecosystem, a healthy human skin ecosystem. This is not a thing that you must hang your head in shame. This is never hang your head in shame about any of the invertebrates that you play host to. This is a gorgeous part, I hope you will agree, of a normal part of human skin. So, they consume cells in the hair follicle or in the sebaceous glands, and they're not very mobile, very reduced. Little tiny, you saw those little bicycling legs? That would be a very strange bicycle for a mite to use. So they only move about 8 to 16 centimeters per hour. And low tolerance of heat and dryness. And this was kind of one of the papers that stimulated us into thinking about this experiment, because when I mentioned that we've known about them for a long time, 1552, but they were still one species, Brevis and Follicularum, until 1972. And then, only in 2015, which is not that long ago, there was a study of mitochondrial, remember the word search, DNA, from a small number of human hosts that they talked about, and they sequenced the DNA of the mites from those human hosts, and they found what they thought was a really interesting combination of host, or an effect of host ancestry on mite divergence. So for one, this is an intraspecific, right? Intra, within species, tree that you're, circular tree that you're looking at of Demodex follicularum in the center there. And you can see deep branches, deep divisions within this one species. For a mitochondrial gene, like this one is, which is called cytochrome C oxidase 3, and we'll talk about that as we go on, COX-3, this is a surprising amount of diversity to see within a species. This diversity suggests that these lineages have been separated for millions of years. Literally millions of years. That's kind of amazing. And so they, the authors of the study were quite excited about it, and they talked about the fact that, well, we can see in Africa where our kind of species emerged into the world from, we see that's where we see the greatest diversity of mite lineages, and as humans kind of moved across the environment, we see a reduction and in the number of mite lineages and a specialization. So all of the lineages that they found, 1, 2, 3, 4, 5 of them in this particular picture, exist in Africa. And then when you get to Europe, most of them are just this clade, they call this pie here in blue, which they call clade D. We'll come back to that a little bit later. So first of all, I gave myself a reminder to check the chat, but I forgot to write it down. Is there anywhere I could find my Mite code? And I see Sherry's already answered that. So yeah, this is because doing this experiment over time has been a real experience for me, because it meant that as someone who works with invertebrates professionally, I'm very familiar with not having to write animal care protocols, because to most universities, animals do not include invertebrates, which you would, of course, I hope, agree with me that this is ridiculous. But because these particular invertebrates live on people, there's a 54-page document that we had to fill out, and part of that was making sure that there was anonymity in the codes and in the process of which sequence was associated with which person. So we don't actually have the way to backtrack whose code who we sent that code to. So that's fine, because not everybody's code worked. It produced a Mite, produced a DNA sequence, so there's going to be lots of opportunity, and as we go along here, you're actually going to see all of the data, and you can choose which one to search. Is the Excel sheet we typed that info in still available? I believe it is. But I'm not going to pull it up right now, because then I would fall over, because that would push me into a critical over number of things all at one time. Since their food supply depends on sebaceous gland proliferation, would it be more likely to find them on people who sweat more? Interesting hypothesis. I like it. So, January was a long time ago. You remember, but again, a quick review of what we did. Sherry, in different years, has posted beautiful photographs and videos of bicycling mites. And what we did was ship you these latex strips. And this was a particular kind of a specific to us attempt a few years ago to make things that we could ship around, and it became very COVID compliant, in that we could pop these in envelopes, self-addressed stamped envelopes, and you put your hands up, pat yourself on the back. We're amazing, because you slept with these fast, you popped them in the envelope, and you got them back by mail or by direct delivery to a mailbox, and we got them all. And so what we did at that point was think about how we were going to answer these questions. Some of these questions we can answer just by looking at who had mites as we look under the microscope, which is in a pre-COVID year, you would have been looking under the microscope. But we sampled these, Sherry and Jenny and Carolyn and Gavin sampled these under the scopes, and they figured out how many mites that we found, the number of mites per host. In different years this year, we didn't, like, what kind of factors were associated with who had mites: the sex, the age of the person, their identified sex, their number of pets, their moisturizing routines. How many mites were we able to sequence? Was it a 1-to-1? If we found something, did we get a sequence? No. What kind of genetic variability was there within the sequence, and is that associated with our ancestry? These were questions we were interested in doing, and as I said, you did amazing. This was the stack of envelopes that returned, and in fact, some of them were even annotated with observations of the mites that they hoped to see. So we can see here an envelope that came back to Sherry that has a mite on it. We can see a picture of a mite from your class here with the code RYTT, with a little stubby bit of hair follicle that your nasal strip pulled out of your head. And there we see the tip of that mite. And you can see some longer mites, more fully exposed from earlier years in these other pictures. So we did find them. Now what we did then was take a little pair of scissors, snip a little triangle out of that nasal strip, and pop it into one of these plates, an 8x12 plate that had a little bit of ethanol in it. And then we took those plates to the Biodiversity Institute of Ontario, which is the home for the Canadian Centre for DNA Barcoding. And they took those little nasal strips with the plastic, with the glue, with all the human hair follicle and the human DNA and hopefully the mite DNA, and we extracted that DNA and we sequenced it for a particular region, for the DNA barcode region. And this year we did something a little bit in addition to that to help us do this big comparison to ancestry. And that's where we left it. That's where we've been basically since January. So now we can start talking about some of the results. And we can start talking about some of what I hope you're excited to see and have some hypotheses about. So if you've got your part of your brain that generates hypotheses kind of asleep, apply some caffeine, because we're going to do some stuff actively across the interpipes. First of all, where did we find them? So this is based on where we found mites as to how you identified sex or gender on your anonymous slide. And so you can see here, of all of the mites that we have sampled, and this is across all 3 years, so your data is embedded in here, we found mites, about 80% of our samples, so that's things that you shipped back to us or that we sampled in lab together in previous years, 80% of the mites that we saw came from women. Now there's a slightly different way to measure that, and part of that could be how many of them sequenced successfully, because sometimes in some years what we thought was a mite might not have been a mite. And interestingly, if we look at that subset of the data where we have DNA sequences from the mites that we thought we saw, you're going to see that we had you're going to see that we had the same proportion. About 80% of our sampled mites were from women. Now, why might that be? I'm sure you have some hypotheses. Why might we have found more mites on female students, female identifying students than male identifying students? So if you could go to Menti and enter into that code 5826 0048, hypotheses about why did we find the mites where we found them. And if you're having trouble, please let me know in the chat, because I think you should have no trouble. And I see some answers popping up, so that's good. So I believe you can get in. Why might we have found mites more on females? That's good. Okay, so I see lots of things. Oh! Oh, I'm going to pull this up. I love it. This is great. So, look at some of these hypotheses as they jump around. Now remember, in this kind of a word cloud, if you use the same phrase, it increases in size. But let's pause this. Oh, forget it. Smith's not going to pause it. But we're just going to talk about some of what's popping up here. And I can see many of you have thought about skin care and more oily skin, softer skin. So hypotheses about the environment that female skin might provide compared to male skin. Skincare routines even. So that's one kind of branch, one hypothesis. Another one that I see popping up a bunch of places is the proportion, and I see this in terms of larger female sample, more females taking their classes. This class, larger female sample, more women in class. And I think that's a really important thing too, because what we have not done is take a look through, I haven't controlled those graphs for the proportion of people who are in this class. So there's two reasonable hypotheses there. And I think we need to collect some more data and have to spend some more time together to fully test why that might be the case. But I would say the hypothesis that I saw jump in here earlier, that men are gross, I can't discount that hypothesis. And let's just leave it at that. Okay. Now, so you answered some more data about the mite. You told us about where you sampled it. And so we've got two ways to measure where you found your mites. And this is data just for this is special data for you in 2021. So some of you who found mites, about 50% of you sampled on your forehead, smaller 25-ish% of you, a quarter of you found it on the cheek, and then a smaller, say 18 to 19% of you found it in and around your nose, which is where mine came from. So this suggests to me that if I'm feeling bad about the number of mites that I have in the future, what I should be doing is taking what I have, which is a large surface area of forehead that keeps growing back until it meets my back, and that's where I should be sleeping with these nasal strips to take advantage of harvesting my mite abundance and diversity. But the same question, is this the same proportion? If we look at the proportion of mites that sequenced, and it's, again, the forehead seems to be a particularly fertile place to find your mites. So file that away for the next time somebody asks you about how mighty your forehead is. One of the last questions we asked you about was the pets. And what we see here, I'm not entirely sure how to look at this, but I split it up in terms of pets in general. And so these are sequences, both of these numbers are numbers of you that have dogs and cats. So if you had yes, you have a dog, yes, you have a cat, yes, you have a pet in general. And the numbers are numbers that had mites, numbers that mites that sequence. So generally, there's two things I think here. This yes column is bigger. So those with pets had more mites, but the proportions, particularly with cats and dogs, which do have another species of Demodex that lives on them, that won't live on us, we don't see any change. But there's an interesting large change in terms of other pets. And so that kind of makes this pattern where if you have pets, we tend to see that you have mites. So let me jump back. Speaking of pets, let me jump back. Yeah. Okay, so I'm wondering if skincare routines like eyebrow waxing or plucking with tweezers affects the population size. That's a great question. And again, something that we could kind of dive into with more time over the years with more students. I guess it really defeated the odds because a female sampled on my forehead and mites weren't found in my sample. Oh, c'est dommage. That's very, very disappointing. But we will continue. Charge on. So the next is thinking about their genetics. So this is, we're going to keep your browsers open and your codes ready. This is where you're going to actually have a chance to see some of the data. And we're going to talk about facial mite genetics, but first we're going to talk and remind you about what it is that we actually intended to sequence and what we've sequenced over 3 years so far. And that's what a DNA barcode is and why it's useful. Now, Jenny's already talked to you in labs about this, but like a real kind of a succinct definition of what a DNA barcode is that it's a short, standardized sequence that enables species identification. So it's four S's, short, standardized sequence. Okay? For animals, this tends to be a mitochondrial gene, cytochrome C oxidase 1, one part of this CO1 gene that's involved in cellular respiration that we tend to all have. There's some. And it codes for a protein, so it's relatively, it does a job, and so it doesn't just mutate willy-nilly and cause lots and lots of changes through time, that we tend to lock it in, and it does a job. And we tend to use this approach because, as you can imagine, for lots of reasons, but one of them is that for many, many things, that don't see the way the world that we do, that don't see it at all in some cases, having some indirect estimate of how isolated populations are from each other, like this DNA barcoding approach permits, gives us a better and a stronger idea of how we can identify things, particularly when their life history stages are very, very different. Think about mollusks, for instance. Think about gastropods. Think about how many kinds of larval type did I talk about when talking about larval pods. The answer, quantified, is a lot. And most of those species of mollusk that have names, that's the adult that has names. Most of those species, we don't know what the larval stage looks like. We know that they follow, in some cases, they follow this progression, but we don't know what the larval stage looks like. So having a DNA barcoding approach where we use DNA to help us name things is going to be super useful if we're interested in naming and understanding the diversity of species that we share the planet with. So it's an analogy you're familiar with, because if you scan that, you understand that's a can of tuna, and if you scan this, you understand it is a tuna. And so, in addition to thinking about this gorgeous mite as Demodex Follicularum, you can look at this string of A, C's, G's, and T's, these nucleotides, and read that in the same way. And you might not be able to read it, and it's hard to pronounce, but this is, to a computer, this is a very readable phrase that says Demodex Follicularum. Now, it says it's Demodex Follicularum if there is a library of data, a database of DNA sequences and named things that help you kind of align and say the sequence of that slide is this named thing. And that was one of the things that we were trying to do by starting this project with all of the invertebrate enthusiasts of Zoology 2700. Was when we started, there were only 4 DNA barcodes, 4 individual mites that existed in the literature, in the world of publicly accessible DNA sequences for Demodex Follicularum. That's kind of bananas. There's billions of us on the planet, so there's billions upon billions upon billions of Demodex Follicularum on the world, and they might be doing some crazy interesting things like following us around through generations. And so we thought it would be really helpful to add to the world's understanding of this group if we sequence this standardized locus for this normal part of the skin microbiome, or the skin environment, let's call it. So that's what we did. And there, the red arrow is pointing to cytochrome C oxidase 1. The mitochondria, this is a picture of the follicularum in the center on a piece of skin. And then you can see all these colored kind of arrows. This is a mitochondrial, circular mitochondrial genome, where different slices, different genes, different regions of genes have been broken up. So COX1, that's the one part of that is the barcoding locus right there. Now, interestingly, what Pallipoli, that genetic study through time, the maps and the phylogenies that I showed you earlier, they use this marker over here. So, since January, what's happened? Well, your mites went into this building on the Guelph campus, the Center for Biodiversity Genomics. And they began to be extracted. Tissues were lysed. The DNA was extracted, and they went through PCR, which you all know about, this polymerase chain reaction. So we add small bits of DNA called oligonucleotides or primers that take a whole morass, all of the genome of all of the DNA of both you, because you're in that sample, and your mite, and they search out one particular part of cytochrome C oxidase 1, so the barcode region of the mite, and hopefully not the person. Then they amplify it, and then we check this out, and we check it out by using agarose gel electrophoresis. And so here on this slide, these are all of those ASDEM numbers. Those are individual wells, so individual mites off of nasal strips from this year. And if you see a little white mark, that's lithium bromide that has bound to the DNA of the amplified barcode region and is fluorescing under ultraviolet light. And we're using it there just as a check to say, has it worked or has it not? And what we found was that of the, say, 80 plus sequences that were sent in, we had successful amplification for about half of them. And this is what that looks like. So they run through a sequencing reaction, and we see different peaks showing different nucleotides, A, C, G, and T. Now, I'm going to give you another kind of a colored way to look at that. And so we've gone in your class, you sent us via the mail your nasal strip, you slept on it, so RYTT, this was your mite. This is actually the DNA barcode from that mite. And so you can see we've got about 600 to 700 base pairs, where each individual color here represents an A, a C, a G, or a T. And that variation, that you have now added a DNA barcode from this region of the circular mitochondrial genome to our understanding of mite genomics. So well done you. Now, you're interested perhaps in who's worked more than just RYTT. There's the numbers that I just said. And I'll tell you who's worked in a second. So 151 strips came back, 81 samples under the microscope, so about 53% of the strips that came back worked, and about 50% of those had successful sequence. These are underestimates. So you could look at this and say, conservatively, half of the class has mites. More than us, probably 100% of us do. This is, both of these numbers, see, these arrows represent a sorting or a filter of how we looked at the world. How we looked at the world in the first arrow was the nasal strip and how well that worked to harvest mites. It's not perfect. Many of these other, it's an adaptation so that we could do this at a distance, pre-COVID and during COVID. And so there are other ways to sample mites that are more invasive, that involve literally sticking crazy glue onto your head and then peeling it off. You can imagine that writing the permits for that is a little bit more intensive. And then, whether or not they sequenced correctly or successfully, that's another filter. So at least 50%, so more than 50% of you have these mites, and for more than those 50%, we have DNA sequences that we've added to the rest of the other years that we've looked at this throughout Zoology 2700 since 2019. So that's a great question I saw just pop up. Do we find fewer mites this year than in previous years? No. That's interesting. So that's one of the things that you've helped us demonstrate, is that mites Not only does the nasal strip work pretty well for sampling mites, we can ship them in the mail. That's kind of exciting as well. So, what did we find? Now this is where I'm going to take a look here. And you can see on the left-hand side there's labels that have to do with which particular mite that is. And I do not expect you to read that. What I want you to look at is colors left to right and top to bottom. Left to right, these are base pairs in that barcode region. Top to bottom are different individuals in our sample. We see a lot of, this is all from 2021. This is all you. This is a lot of individuals that sequenced successfully. And what I'd like to do is with you, I'm going to put a link in the chat. That link is going to take you to a Google Doc where you can take a look at those sequences. So get into that Google Doc, and this is what I want you to do. I want you to Control F or search for your code. If it's not there, if it's there, bully for you. If it's not there, just choose one of the sequences. And what I want you to do is to highlight the sequence and copy it. And then we're going to use two different identification engines, and the links to those identification engines are in, you can choose, doesn't matter which one you choose. But the links are in that Google Doc. So you just click on that and you go. So it'll look somewhat like this. You'll go into that document and you'll find a sequence. Let's start that again. So you go into the document, you find a sequence, you copy it, and then you click on, let's say, so copy that, then you click on either the blast engine or the barcode, the bold systems. That'll open up another window, and you paste that in. And then you click either submit if it's bold, or it says in the sheet, I don't remember what it says for blast. But then you're going to get a result, a sequence result of what that is. So I see that there's a bunch of you in there. So this must be working. Now, what I want you to do is, if it is working, what I want you to do is use this new Menti code and paste 35325583 into Menti, and I want you to paste the identity of what that sequencing reaction told you. So what you I want you to read the sequence, and I want you to share the result in that Menti. So I'm still looking at here. I don't see any questions yet. So I'm going to presume that you're successfully sequencing. And I see some results coming up. Good. It's working. Now, you can share this with the rest. Share with the rest of the people in the chat, the rest of your colleagues, what you're finding. Yeah, and you see, so with an S or without, there's not a lot of variation in what you're finding. You're all finding the same thing. Could you repeat how to paste the sequence? Yes. Let's go over here. So, if you can see this screen now, you copy it, and then you click on one of these links, and you go to the link, and then into that link, you paste it, and you say submit. And then you wait. Maybe you drink your coffee. Maybe if your hands are moving like this, you don't drink your coffee, you drink your tea. And then you get a result. And this says, yeah, so you're seeing what I just did there. 100% Homo sapiens. Now, what does this tell us? Hop over to the chat. Menti code is 35325583. So give me some ideas what this is saying in the chat, and I'll go back to what this looks like. Yeah. Z. Faulkner, Carolyn, you're exactly right. So is that our DNA barcode? I.e., is that the human DNA barcode, not the mite's? Bingo. So, remember that DNA extract, you're going to have to tell me what cyan is. I'm not sure what that is. Why is some of the map cyan colored? Use simple words, small words, little tiny shiny brain. The color blue. Oh, the color blue means that's the adenine, guanine, cytosine, thymine. This is one particular nucleotide. So there's not a lot of it, but there's so we don't have an equivalent number of the 4 bases, and that's a common thing to see in arthropods. So you were right with your hypothesis. The human DNA is mixed up with the mite DNA. So is that an error? Well, in fact, that to me, let me just jump back ahead to where I was here. What does it mean that we found DNA? I don't think of that as an error because we know we didn't try and exclude the DNA. The DNA extract that's in there is a mixture of the mite DNA and the human DNA. So this is one particular run that was kind of a happy accident that was completed in the lab where they sequenced too fast and according to the wrong protocols. And they used primers that were designed for vertebrates, us, as opposed to primers, these bracketing kind of bookmarks that sit on either end of the region that you want to amplify in the polymerase chain reaction, that would be designed for arthropods or for Demodex or for mites in particular. So this was a happy accident where we get now to look at, you've now sequenced your own DNA barcode. And one of the things that's interesting to me there is that we had much greater success. So we know that those nasal strips, you did sleep with them because they have hair follicles on them, and there you go, a gift of your own DNA barcode. So the next time that you see your mom, you can say thank you because that's a maternally inherited gift, and so that's her DNA barcode as well. Now, that mistake, that happy accident, so I'm not going to call it an error because those were primers that I didn't ask for but were designed to go after vertebrates, and so they found the vertebrate DNA that's in that slurry of DNA extraction. What about the invertebrate DNA that we were more interested in? Well, we got those as well. So this is what they look like. Now, the second sequence, what we're going to do is I'm going to share a second Google document with you. Here it comes in the chat in a second. Do the same thing here. And first of all, think about, I see a different pattern in the variation I'm looking at. I would imagine you think that. So what are these sequences that you're going to answer that using the Google document? And think about maybe how many species are we looking at there. So go to that second Google document and do the same thing. Search for your code and paste it into one of those 2 search engines and send me back what your results look like by going to Menti again. Oh, it says we need access. Well, how rude is that? What about now? That might have just been the way that I was. Try it now. You should be able to see it now. Apologies. Works now. Hooray! Good. So take a look for your code, and if you don't see it, that's fine. And there's lots of other codes. This is actually all of the DNA for all of the years. You can take a look through there. Pick one of them. Go into one of those other links, and let's take a look and see what you get. And I'm going to ask you, once you've done that, to go back to Menti and I think it's the same code. and put in your sequencing results there. I can see you in there, so that's great. It's working. All right. All right. Now, those of you who put in, I see that some of you are finding no match. Which database, if you found no match, tell me in the chat which database you're using, if you could. It's not that many of you, because there's what we're seeing. Most of you are finding this is your facial mite. So let me help with this introduction. Facial mite, you, the DNA of your facial mite. Hooray! That's amazing. I think this is amazing. This project, I'm so glad that you agreed to participate in it, because this is kind of exciting. We've gone from talking about serial endosymbiotic theory back in January to mailing you nasal strips to sequencing the DNA of the mites that lived on you, and now being able to compare and contrast the variation in your own barcode to the variation within the intraspecific variation in your facial mite. And that's kind of the next question that I want to ask you about. And I see, yeah, some of you are finding no match. That's actually, you can go back and try that again, because there's a setting in bold that lets you, you just want to increase the number of the proportion of the library that you're searching. And you can go back and do that later on, or I can tell you about it in the chat after lecture, but it's an easy, just, you make the search criteria wider, and then you'll get the match. So, what I'd like you to do now is actually to, in the chat, if you could, cast your mind back to what that sequence, that screen grab of all the colors with cyan and other things for the barcode region for you, the host, and compare and contrast it to what your mite DNA looked like. Who has more variation? Why might that be? What does the graph represent into the bold output? Yeah, it's the confidence, it's the number of things that it bumps into. And so in most cases, like with your human, there's a lot of human DNA, a lot of human mitochondrial DNA in that database. And so for this region, it's pretty darn confident that that is what it is. There's very little ambiguity about what that react. Okay, so yeah, so I see maybe what you're asking for here is, I'm going to jump back and say, this is what all of your mite sequences look like as a picture of a color. This would be a great scarf. I would love this as a t-shirt. This is what the human DNA looked like. So who has more variation, and why might that be? So the host, the vertebrate, and the invertebrate. Who has more variation? Pop that into the chat. I see some answers coming up. I'm going to zip it for a second, just so to give you a second to open up. I love this stuff. All right. So let me share some of this. You're seeing it, but I'm just so that we get it on the video for later for the people who are attending asynchronously. You're right, it's the invertebrate. And the invertebrate, so is it the invertebrate because there are more gray lines? The invertebrate has more variation. So it's not the more gray lines, it's actually the variety of colors. And it's the frequency with which the colors are changing, not from left to right, but top to bottom. Left to right is the sequence within an individual. Top to bottom is the sequences from different individuals. So as you see colors across those vertical lines kind of changing, that's showing you polymorphisms at the sequence level within one species. Isn't that cool? So why might that be? Maybe because they diverged millions of years ago compared to Homo sapiens. That's a super good observation. These are old lineages. Their divisions predate us, but now they live on us obligatorily. So what would have Neanderthal mites look like? We need to know. More variation because they have a shorter lifespan, because there's more turnover. That's fantastic. That's a great hypothesis. Larger population size, and they're smaller. So again, thinking about how quickly they turn over. that different mite species have been separated for a long time. That's exactly right. So much longer. These are super good hypotheses. I really like how you're interpreting that, and you're correct. So the mites have a lot more variability than we do as a species, and that's a happy accident that we get to look at directly at your DNA and your mite DNA in the same term. Amazing. I love it. Now, what did we see when we looked at it? Well, I'm going to slow myself down and take us... No, I'm not. This I'm going to hold. Don't worry about doing this. We're going to do that later on. And I'm just going to tell you that there were actually two other, if you take a look at those colors, no, I'm not, yeah, blah, 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 blah. He jumps through those last three slides so that we have time to talk about the geographic patterns. Now, one of the things that I wanted to tell you about, particularly in other years, when we had time to search through the nasal strips with greater care, because you were doing it and not us doing everybody's, is that we found a lot of cases where individuals, the host, the people, the students, had multiple mites. That's maybe not a surprise. It's not just that we have one. But these are multiple mites that have different haplotypes, so different DNA sequences. And so 3/4 of those had multiple, of the people that we found had multiple haplotypes for individuals. And what might that look like? So you can see here the anonymous code WWQT, and we have two different mites that were pulled out and sequenced, and you can see there's a synonymous transition from a C to a T, a C to a T, a C to a T, or a T to a C. So there's variability. So you don't just have one lineage of mite potentially on you. Potentially you have multiple mite lineages. That's amazing. Now, the last thing that I wanted to talk about in the last 5 minutes was the Pallipoli paper that Megan Thames and others and Rob Dunn, who've looked at kind of mite divergence on humans, that stimulated us to look at into this as a project. They use mitochondrial DNA as well, but they use cytochrome oxidase 3, not 1. So not the barcode region, but a separate region. And this is kind of an example of like a Tower of Babel, where we can't necessarily compare our results to what they found unless we amplify that same marker. And so what I did this year, because time or opportunity or just stubbornness, is that I designed new primers that we could use to overlap with what they used for a small snippet, and we could sequence our all of the years that we've done this and add. So we've taken this now from nasal strips to searching under the scope to sequencing the barcode region to now sequencing a snippet of the same region that Pallipoli looked at. And that lets us compare your results exactly to what they found. And I think that's kind of cool. So this is a reminder of what they found. Now, there's the phylogeny in the bottom, so deep divergences within the species. You've seen that, and you've seen it within the barcode region for the things that we saw. So that's kind of the same pattern. And that's to be expected. These are both mitochondrial markers. The mitochondria is inherited as a unit, and so different genes may mutate at different rates, but we wouldn't expect to see different patterns within that replicating circular mitochondrial genome. Now the map is kind of the pattern, the geographic pattern of hosts that they saw. So remember, we kind of anchor it on Africa and say, in Africa, where humans come from, we see the majority of mite lineages. And then as we hypothesize, as we see different migratory routes and finishing out human populations where we self-identify our ancestry, we see different strains or different lineages. So here, this is there on the outside of this phylogeny, that's their big clade D. And that's here what predominantly lives on those who've identified as being of European ancestry. Those who have identified as African descent of ancestry, there's a lot of different lineages, including D. There's another haplotype named B and A that tend to predominate in those of Asian descent. And then in Latin America, in South America, and Central America, different haplotypes, the A haplotype becomes dominant. So that's what they found. Now, what did we find? This is a different way of looking at it. You can go and look at these phylogenies yourself and search your own code if you want, because you can't do it on here. If you go, I'll put that link in the chat here if you're interested in doing that. Or you can just scan it with your phone, because it's the same thing there. basically, if you take a look at the clade on the outside, so that's me identifying your sequence based on where it falls into comparison with the Pallipoli data. So most of what we found is clade D. We found some small representation of clades A, B, and C. And one of the cool things is this group down here at the bottom that some of you are part of. This is a new lineage that we haven't seen before, that has become evident in the sampling that we've done in this class. So not only have we learned something and been able to compare our data to that data that was published in PNAS a few years ago, we've discovered things. So what does it say in the last minute about the host lineage? This is what the Pallipoli data showed. This is what your data showed. And this is kind of interesting to me. Clade D is everywhere. We see clade D everywhere. We don't see the same diversity of mitochondrial lineages emerging from hosts identifying of African descent. We still see Asian ancestry, the hosts who identify of being of Asian descent having clades B and D. And the new clade also seems to be associated with European ancestry. So we've asked a lot of different questions in a very short period of time. And you're going to have a chance to dig into these results more, because there is an assignment associated with this data that you're going to have an opportunity to do together in lab this week. And so you're not done taking a look at this data yet. But what I want you to do as you think about it is reasons that we might not have found the same genetic diversity in hosts of African descent. What might that suggest? And what I'd like to do then, finally, because I'm looking at the chat, is tell you about the things that we've learned. We're going to tell you about that in lab, and I'm just going to end with this, that this is a quote from your text where they do talk about Demodex in several cases, but with a mixture of reluctance and relief, we leave the mites. You're going to leave them shortly after your lab. And I want to say thanks to all of you for participating in this. This is kind of a unique opportunity that we've developed here at Guelph, and it's hats off and kudos to you to helping us do it and conduct this experiment together during COVID. So well done, you. I'd like to keep talking about this for a long period of time, but I realize that you have other places to go and probably other classes to think about. So I'm going to stop recording and say thank you, and take care of yourself, and take care of those that you live with, and stay physically distant, and you'll see more of your mite data in lab this week. Well done, you.